Hello everyone, I welcome you all to the third lecture of this series related to coronavirus. In this video lecture, I will talk about the evolution of these viruses to become human pathogens and how they jump from their natural animal host to humans and why human immune system cannot cope with this new strain of virus that has never been seen before by the immune system. How do SARS-CoV-2 is causing the COVID-19 pandemic? Fits in the context of other viruses that are circulating within the human population or have caused previous epidemics. Because we as a species are always being exposed to viruses, as is illustrated very nicely in this image, how humans are being chased by all sorts of viruses and pathogens. So using this perspective, I would like to address three questions about SARS-CoV-2. First, how do viruses such as SARS-CoV-2 enter the human population? Second, how does SARS-CoV-2 relate to other circulating and epidemic human viruses? And lastly, based on other human viruses, what might we expect for long-term immunity or coexistence with SARS-CoV-2? So first, it's important to point out that every human pandemic virus that we know of in recent times has originated from another species, which is something we call zoonotic transmission or zoonosis. I am showing here a case in which we are saying it's coming from a bat, which is called a reservoir species. For SARS-CoV-2, bats were likely the original reservoir, but of course many different animals serve as reservoir species for zoonotic viruses as well, as we'll discuss in a moment. But the zoonotic transmission is a single human is only the first step. There are also a very important second step, which is the virus that needs to be able to have sustained human to human transmission. To these two steps together really result in a virus that has pandemic potential. Now we look at this in the context of coronaviruses. We know there are plenty of circulating human coronaviruses that cause mild symptoms that we often refer to as common cold, and up to about 20 years ago people didn't really pay too much attention to these viruses because they were just one of many types of viruses that cause a common cold. What we have also learned in the last few years is that the fact within animal population, again especially among bats, there are many circulating coronaviruses. And again we assume that because these are resonate in these animal population, these have pretty low case fatality rate. So where the danger comes is when infected bats or some intermediate host comes in contact with humans, and we have what's called a spillover event. These spillovers result in zoonotic viruses in the human population, but fortunately many times these have limited or no real ability to transmit human to human. And within the coronavirus family, we have an example of this where starting in about 2012, we started seeing cases of viruses known as Middle East Respiratory Syndrome coronaviruses or MERS-CoV. We have been about 2500 cases of the virus, and as is common in these type of zoonotic viruses, the case fatality is really quite high, which is pretty alarming. But again, human to human transmission appears to be low. But for other viruses, once that spillover event occurs, either because the virus was initially adapted to do it or rapidly adapted to do it, now it's able to have sustained human to human transmission. So these are the viruses that have massive pandemic potential, and this is where we are with SARS-CoV-2. In this case, the very first case appeared to have been detected in November or December 2019, and as of April 22nd, we are rapidly approaching 2 billion cases globally. What we also see is that the case fatality rate is much lower than MERS. There are early reports also of this occurring in coronaviruses, where in 2002 there was an outbreak of a virus that was known as SARS-CoV, and fortunately it was stopped before it spread globally, but was also quite deadly. So we already know that pandemic potential in the family of viruses, but of course SARS-CoV-2 has really emerged on a much larger scale. So if we want to understand how this happens, we really need to understand the evolution of viruses and hosts at a molecular level. So what leads to the emergence of pandemics? So it's important to not just think about the species, but actually the viruses within those species. And as we have already seen in last lecture, viruses mutate, and so that we know that the virus that's circulating in human is only about 5% different compared to known circulating bat viruses. And if we want to know how many different differences change the virus, we need to think back to the life cycle of the virus. All these points of contact shown here in the schematic diagram between the virus and host along the life cycle can be barriers that the virus needs to jump to get into the new population. The one I am going to focus on now is step by which the virus enters the cell, which is binding to the cellular receptor, which is mediated by an interaction between the viral protein that's called spike protein and the host cell protein called ACE2. And we often schematize these interactions as basically a key that needs to fit into a lock, but of course the real interactions look much more like this, with a three-dimensional structural interaction of the spike protein that then interacts with this ACE receptor. So now if we go back to our bats, we know that the bat virus must have had a spike protein that could interact with bat ACE2, but a circulating bat virus may not necessarily be able to interact with human ACE2, which it would need to be able to do to jump species. And I have drawn ACE2 as looking different because we know that host proteins that interfere with viruses tend to themselves evolve very quickly across host species, presumably because of these high stakes host virus conflicts, and virus mutate a lot. Some imagine that within the bat population, a variant of this virus arose that could utilize human ACE2, and if that right virus encountered a human virus, they could transmit it to humans. So the final thing to say about this is to just reiterate that spike and ACE2 were only one piece of this puzzle, and for a virus to be successful, it needs to adapt to many of the genetic differences between humans and the reservoir species. So for instance, we already know that there are many coronavirus circulating in bats that can already utilize human ACE2, but presumably haven't made the jump into human because there are some other molecular barriers to replication. So, having talked about coronaviruses such as SARS-CoV-2 can and have entered the human population, I want to return to the question of how SARS-CoV-2 relate to other circulating epidemic genome viruses. As I mentioned earlier, there are coronaviruses that span this whole range of steps in human viral emergence, from animal viruses to zoonotic viruses to circulating human viruses. But as we know, many human viruses and have had many human pandemics. For example, one of the common things we are hearing about how these are related to influenza virus. Partly that's because the influenza virus caused respiratory symptoms like coronaviruses, but partly there's also just a very clear analogy here in terms of the variation influenza viruses in the categories shown here. So the pandemic level, we have all heard of these major pandemics flus from 1928, the so-called Spanish influenza, and even as recently as 2009. But of course, we know that in addition to these pandemic strains of flu, there are several seasonal flu strains of influenza that we have to deal with every year, as well as strains of viruses that transmit from birds and have heavy case fatality rates, but so far have limited human-to-human transmission. And as with MERS, one of the big concerns is that if any of these viruses like H7N9 bird influenza virus gain human-to-human transmission, we really need to worry about that. And we also know that the real reservoir of the viruses in the many strains of avian and swine influenza that circulated within an animal population. And at the same time, there are many other pandemic viral strains outside of influenza, many of which comes from some animal reservoir at some point. So, for example, smallpox and HIV and Ebola have all caused epidemics in humans. And we know, for instance, that HIV transmitted into the human population several independent times from primate reservoirs, only a little over 100 years ago. Of course, we have plenty of circulating human viruses like measles, polio, that probably had some zoonotic transmission deep in them, in their ancestry, but wasn't quite as recent as any of the as well as virus in their category of zoonotic transmission with high case fatality. Among these is rabies virus, which is 100% fatal if left untreated, but doesn't transmit person to person, and also have Nipah virus, which has a very high case fatality rate and is also famous for being the virus people used as the model in the movie Contagion. And finally, this is where some basic virology surveillance has taken place. We now know that there are many viruses circulating in reservoir species that have an unknown number of evolutionary steps away from being zoonotic transmission. So one thing to confront is about all these other viruses that we aren't constantly dealing with influenza pandemics and smallpox and other pandemic viruses, and that's because of the largely effective role of our immune system in dealing with these viruses. Once the immune system has actually been prepared, and so with that I will start talking about this last question of what we might expect for SARS-CoV-2 in long term. It is too early to know about long-term immunity of SARS-CoV-2, as all of this information is only recently emerging. But we can get a hint from some of these viruses that we have long-term protective immunity against many viruses like smallpox, rabies, measles, mumps, rubella, and several others. So we have really good ways of making vaccines, and the hope is that this will hold for SARS-CoV-2 as well, although the development of vaccines take much time. We also know that in case of something like Ebola, where we didn't yet know we have a good vaccine, but we know we can take blood from people that have been infected and then cleared the infection, and use antibodies from that infected person to administer to people that are eventually infected. This can actually be quite protective. So the SARS-CoV-2, we expect this might be a limited but effective way to treat current infection. Of course, there are several viruses which we have limited short-term or unknown level of protective immunity. Unfortunately, for instance, one thing we don't know, because circulating coronaviruses are not incredibly well studied. We don't actually know whether people have long-term immunity to these common cold coronaviruses. Some work actually suggests that there can be short-term immunity, maybe a year or two, but people can eventually be reinfected and essentially the same strain of coronavirus. So this could have implication for what we could expect from SARS-CoV-2. Also, we know that we need a new vaccine every year against influenza virus. This has less to do with how effective the vaccine is and more to do with the vast rate of influenza evolution. The upside here is that even with limited immunity, but because of viral evolution, we know that the pandemic strains of flu with high case fatality rate don't endure right. They essentially turn into seasonal flu in future year. Based on this, I think overall this is encouraging, in the precedent with the other viruses suggests that once we can get in front of the virus on the public health level, we might expect effective we might expect effective protective immunity against SARS-CoV-2, and while we don't know yet what the long-term future holds, many of these other viruses can be contained by either effective vaccines or protective human immunity. So these are the references that I consulted for making this video lecture. I just hope you like this lecture. Please like and subscribe to my channel. Thank you.